We're going to shift gears and talk about, I think, a little more look at the future. We're going to talk about long-acting formulations and devices and the potential for those products to have an impact on the treatment of children and adolescents. Let's see, does this work or do we have to use this guy? Maybe this? Yeah, that's it. So here are my disclosures as required by the conference. So I'm going to start with this question. Why is this topic important? Why is it important to be talking about new formulations and drug delivery methodologies for infants, children, and adolescents? Well, it's important because adult formulations of existing antiretroviral drugs are often inappropriate for children and adolescents and/or are difficult to take. Long-acting and extended-release products, on the other hand, have been proposed as an important solution for those having difficulty adhering to daily oral medications. Formulation development for infants and children is an important barrier to manufacturers and to the supply chain for these special populations. And in particular, oral formulations, generally speaking, have to be customized for pediatric populations. You can't use an adult formulation for most infants, in most circumstances for infants and young children. And so this is an issue for generic manufacturers who have to set up a very expensive manufacturing facility for what often turns out to be a very small market, which, needless to say, they don't like doing that. On the other hand, injectable, implantable, and transcutaneous formulations might be simpler to deliver at scale using the same product developed for adults. So, for example, with long-acting injectable cabotegravir and rilpivirine, which we're going to talk about in a few minutes, rather than having to create a pediatric formulation, you would simply take the adult formulation and deliver it to children at a smaller dose. And so companies that say, we can't do this because we can't afford to develop a formulation for your pediatric population are no longer going to be able to say that. And I think that's a good thing. Finally, and most importantly, infants, children, and adolescents have exceptionally poor persistence on oral antiretroviral therapy, and they are the only populations that continue to die in large numbers from AIDS-related complications. I've been going to meetings like this for 2 decades, and we keep saying we're going to have better treatments for children, better treatments for children, better treatments for children. It's time to stop saying that and do something about it. And I think long-acting formulations, these formulations, I think, may give us a window to do that. Okay, so let's talk about what's in the pipeline for HIV treatment and prevention. And we'll start with drugs that are probably familiar to very many of you, injectable cabotegravir and rilpivirine. I'm going to quickly review the phase 3 results of studies of these 2 drugs that were presented at CROI in March. The first is the Atlas study that was presented by Sue Swindells. This was a study in patients who were already suppressed on a combination antiretroviral therapy and were randomized to continue that daily oral therapy or to receive a 4-week run-in of oral cabotegravir and rilpivirine followed by 44 weeks of injectable cabotegravir and rilpivirine. And what was presented was the primary virologic endpoint at week 48. So perhaps not surprisingly, at week 48, the oral arm and the injectable arm were comparable, comparing either the primary endpoint, which was the proportion of patients who were not suppressed, or the secondary endpoint, key secondary endpoint, which was the proportion of patients who were suppressed using a 50 copies per mil cutoff. The one, I think, surprising thing about the Atlas study was that there were 3 treatment failures on study in the injectable arm. And these were not non-adherent patients. These were patients who were getting their injections as prescribed on time, and that would be every 4 weeks, plus or minus 1 week. But according to the presenters, these 3 patients were quite adherent with their treatment, and they failed therapy anyway. Now, in Atlas, which was treatment-experienced patients, most of these failures were failures with rilpivirine resistance. But there was one failure who also had cabotegravir resistance. And there were some interesting features to these failures. The first is the presence of this L74I polymorphism in the integrase gene at baseline, and also the fact that these patients had concentrations of both cabotegravir and rilpivirine that were in the lower quartile for the population on study. Now, the Flair study was a study in treatment-naive patients presented by Chloe Orkin. A similar design, although all patients had to receive combination oral dolutegravir, abacavir, and 3-TC for 24 weeks prior to randomization and had to be suppressed at the time of randomization. And it was randomization to the same arms as in the Atlas study, continued oral therapy or the all-injectable regimen with a 4-week oral run-in to make sure that there was tolerance of the cabotegravir-rilpivirine combo. And again, at 48 weeks, not surprisingly, the two arms were equivalent with respect to both the primary endpoint and the key secondary endpoint. And again, surprisingly, there were 3 treatment failures on study on the combination injectable arm. And this time, 2 of these 3 failures were, 2 of these 3 had rilpivirine resistance, but all 3 on the treatment-naive study had cabotegravir resistance at the time of failure. It's also interesting to note that 5 of the 6 of these failures were from Russia, but there about 17% of the patients on these 2 studies were from Russia, so that in and of itself was not particularly unusual. And there's speculation about why these Russian patients may have been at greater risk of failure despite adherence with the injectable regimen, and that's something maybe we can talk about at the question and answer session at the end. We're also going to be hearing more about this this week in Mexico City, follow-up data from these 2 studies, as well as some more data on the patients who developed resistance. Now, one of the important things to note about this all-injectable regimen is it is surprisingly well tolerated, despite the fact that it requires a 2 milliliter injections in the buttocks every 4 weeks, or in the case of their phase 2 study and some ongoing phase 3 studies, every 8 weeks. And despite that, patients are quite happy with this injectable regimen. And here are the data from the phase 2 Latte 2 study looking at patient satisfaction by asking patients 2 questions. First of all, at the top, how satisfied are you with your current treatment? And at the bottom, how satisfied would you be to continue your current treatment? So it's week 48 data and week 96 data. And the first 2 columns are the injectable regimens every 4 weeks or every 8 weeks, and the last column is the oral comparator. And it's a little bit difficult. This is not the kind of data we're used to seeing, so I'll walk you through it. The pink bars represent the proportion of patients who were completely satisfied with their injectable regimen. And as you can see, on all 3 arms, the majority were completely satisfied with their regimen, and the one exception is the oral people did not, more than half of them were not completely satisfied with the idea of continued oral therapy. The aqua color is the proportion who were extremely satisfied with their therapy, but not completely satisfied. And as you can see, if you combine completely satisfied and extremely satisfied, more than 95% of the participants in the 2 injectable arms were happy that they were receiving this therapy and would be happy to continue it. Now, there's bias in these data because these are patients who volunteered to be randomized 2 to 1 to every 4-week injections every 8-week injections, or continued oral therapy. And most of those people obviously wanted injectable therapy. But even with that, I think it's extraordinary that over a 2-year period of time, despite volunteering for these injections, they still like them and would be happy to continue them, despite what I consider a very large volume and potentially painful injection. But for the people participating in these studies, that seemed way better than continuing to take daily oral therapy. So cabotegravir and rilpivirine have been submitted for regulatory approval in the United States. Regulatory approval is expected in 6 to 9 months. One caveat is that the drugs will be approved with a requirement for a 4-week oral run-in to assure tolerability prior to injection. And in addition, these drugs will be approved initially at an every 4-week regimen because that's how the phase 3 studies were done. There's good data on the effectiveness and tolerability of every 8-week therapy with these 2 drugs from Latte 2 and from ongoing studies, but that's not yet in the regulatory package. So these drugs will come to market as every 4-week injections. Both drugs are very well tolerated. And surprisingly, there is resistance to these drugs that's rare, but it does occur. 8 cases of resistance total in phase 2 and phase 3. It's always seen in subtype A virus, which is a virus that is most prominent in East and Central Africa and in Eastern Europe, especially Russia. And so there may be something about about the ability of subtype A virus to develop resistance to cabotegravir that was unexpected. In addition, all of the patients who failed had the L74I polymorphism in the integrase gene at baseline. That was not previously considered a resistance mutation, but we may need to take a closer look at that. And finally, all of these patients had cabotegravir and rilpivirine concentrations in the lower quartile, so that may have played a role as well. However, the investigators pointed out that there were plenty of patients with all 3 of these conditions, subtype A virus, L74I, and low concentrations, who succeeded with therapy. So having these three things does not guarantee that you're going to fail injectable therapy. It just somehow makes you more susceptible. And obviously, we have a lot to learn about that, and we have to decide what we're going to do about it to try to prevent resistance in the future. I should also say that both Atlas and Flair have been submitted for publication, and so hopefully they will be, the full publications will be available very soon. Okay, I'm going to move on now from cabotegravir and rilpivirine, very familiar products, and I'm going to talk for the rest of my time largely about products with which you may not be as familiar. These are products that are much earlier in the pipeline. So in terms of injectable drugs, let's talk first about the Gilead capsid assembly inhibitor, GS6207. So what's a capsid assembly inhibitor? Well, as you probably are aware, in order to produce infectious particles, HIV has to produce a GAG polyprotein that's cleaved into structural subunits that spontaneously assemble underneath the cell surface and come together as the virus buds out of the cell to form a mature infectious virus. If you can block that process, you can't make infectious virus particles, and you effectively stop the HIV life cycle. There's never been a drug available in this class until now, and so this is quite exciting. The drug has a unique mechanism of action and a unique resistance profile. It does not have cross-resistance with other antiretroviral drug classes. It's exceedingly potent. It has an EC50 of 50 picomolar, and it is possible to select for resistant variants to this drug in vitro, but they have very low fitness because they don't assemble very efficiently. The drug also has low inherent in vivo clearance, and that gives it an extraordinarily long half-life in humans of 30 to 43 days. Remarkable pharmacokinetic profile and really a first for an antiretroviral drug. And it's given as a subcutaneous suspension. So unlike cabotegravir and rilpivirine, which have to be given intramuscularly, this drug can be injected subcutaneously, and that raises a lot of possibilities, especially for populations like infants and children, where intramuscular injections might be more difficult. These are the pharmacokinetic data from the healthy volunteer trial that was presented at CROI in March by the folks from Gilead. And this is looking at the proportionality of the pharmacokinetics over 24 weeks. So this is a 6-month profile after a single injection of this drug in human volunteers. And this is comparing 4 different doses, 450 milligrams and 300 milligrams at the top, and then 100 milligrams and 30 milligrams at the bottom. And doses of 300 or 450 milligrams produced concentrations of this drug above the EC95 for 24 weeks. So this could be potentially an every 6-month injection. And in addition, the 100 milligram dose produced concentrations above the EC95 at 12 weeks. So the potential for an every 3-month injection at the lower dose. So a very exciting drug. We're going to hear the first 10-day antiviral activity data from this drug presented this coming week at the IAS meeting. So please pay attention. I think it's going to be very exciting data and a new avenue, in this case, an avenue that may be very relevant to infants and children. Okay, I want to mention broadly neutralizing monoclonal antibodies. We don't tend to think about these drugs as being long-acting, but they really are. And the LS modifications of these drugs, like GS6207, in many cases could be given as infrequently as once every 3 months or once every 6 months. In addition, they have been used subcutaneously. And that, again, I think opens up a lot more flexibility of dosing for populations like children, infants, and adolescents. And these are data published last year looking at the pharmacokinetic profile of the long-acting version of VRC01LS at 3 different doses and 2 different routes. So at the top is 5 milligrams per kilogram given once subcutaneously in the colored symbols. 5 milligrams per kilogram IV in the red, 20 milligrams per kilogram IV in the orange, and 40 milligrams per kilogram IV in the green, as compared to the standard non-modified, non-long-acting version of VRC01LS given in the same route and at the same dose every 4 weeks in the black symbols in these 4 squares. So clearly, this is a drug that has a pharmacokinetic, the time scale, again, is in weeks. Out to 24 weeks. This is a drug that maintains antiviral concentrations for more than 6 months in human volunteers, and I think opens the door for another way to provide combinations for long-acting treatment. I will say there's going to be bad news about this antibody, VRC01LS, presented at IAS this week by Pablo Tebas, who will give the results of the ACTG 5378 study, which compared VRC01LS to VRC07LS in treatment-naive patients, and the antiviral activity of VRC01LS in those treatment-naive patients was quite disappointing. And so I think this drug is no longer, this antibody doesn't have a future in all likelihood, and it's going to be replaced by VRC07 and probably by other antibodies in development. So another presentation to pay attention to this coming week. Okay, I want to move now and talk about devices. And so we'll start and talk about implants. Implants have been around for decades. They are one of the most popular ways to provide hormonal contraception in the world. So these are not crazy science fiction ideas. These are drug delivery devices that literally millions of women are already using. So the question is, could this kind of drug delivery technology be applicable to HIV treatment and prevention? And the answer certainly is yes. There are a couple of antiretroviral implants that are in early clinical testing. We're going to hear about one of those this week, the Merck MK8791, which I'm going to talk about in a few minutes. But let's start with tenofovir alafenamide. This is a diagram of an inert silicone-based tenofovir alafenamide implant to be delivered subcutaneously. This particular device has this silicon shield, if you will, and a core that is pure crystallized tenofovir alafenamide. And tenofovir alafenamide, being a highly potent drug and one with a reasonably long half-life, gives you the possibility to deliver drug out of a device like this over several weeks, if not several months. This particular device has been tested in laboratory animals. These are data from beagle dogs that were published in 2015, showing the concentrations over time. In this case, the x-axis is days, so this is out to 40 days for tenofovir, the parent nucleotide in the plasma down here at the bottom. Sorry, this is TAF at the bottom in the plasma, and then tenofovir in the middle. And then up at the top are tenofovir diphosphate concentrations, the active metabolite of this drug in peripheral blood mononuclear cells, suggesting that this implant could deliver effective antiretroviral concentrations of tenofovir for more than a month. And this is a device that is currently in early phase 1 clinical testing, and I think we'll be hearing data about this soon. I should mention there are several other tenofovir alafenamide implants in preclinical and even in early clinical development. Those are bioerodible or biodegradable implants. And so this kind of a device is implanted and releases its drug and then has to be removed. And that has its advantages and disadvantages. If a patient develops toxicity, it's a lot easier to remove a device like this if you need to remove it. The injectable bioerodible implants often form a little depot, a little lump. That lump could be removed, in theory, during the first few weeks to few months after it's injected, but it starts to break down, and after a time, you won't be able to remove it. And again, that has its advantages and its disadvantages. Eventually, those kinds of implants can't be removed, but the good news is you don't have to go back 6 months later or a year later and remove them. And we do hear stories about women, particularly in low-income countries, who get Jadel or etonogestrel implants, which is levonorgestrel implants, which is a 5-year implant. And after 5 years, they think they're done, and they're walking around with these implants under their forearms for years. And so that's not probably a good outcome either, although there's not a lot of complaints from the women who have received those kinds of implants. But that's just something to think about for the future. Is it better to have an implant that can't be removed or an implant that can be and must be removed? The other implantable antiretroviral I'm going to talk about is this one, EFDA or MK8591. It is a nucleoside analog being developed by Merck. And we're going to hear this week about the first human pharmacokinetic data with an EFDA implant. So again, something to pay attention to at the conference this week, a very important and exciting development, I think. So this drug is somewhat unique. It actually is unique in the portfolio of antiretroviral NRTIs in that it has a 3' hydroxyl group, unlike all of the other antiretroviral NRTIs. And what that means is once it's phosphorylated, it actually gets incorporated into the viral RNA-DNA duplex. And the way it stops HIV replication is because of the size and chemistry of the molecule. It's like throwing a wrench into a gear. It causes the entire replication machinery to come to a halt. And that may have some important implications for its pharmacology and its resistance profile. So rather than being a reverse transcriptase inhibitor, it's being called a translocation inhibitor because that's its molecular mechanism of action. It's an exceedingly potent drug, and data suggest that it will be possible to dose this drug at less than 5 milligrams per day in humans. It lacks cross-resistance with most NRTIs. The only cross-resistance is with lamivudine. There is minor impact of M184V, but this drug retains activity as monotherapy in primates given virus, given SIV, that has M184V. So despite the impact of M184V, that impact is so small, you can still treat through that at least if the primate has M184V, suggesting this drug may even be useful in humans with M184V, and that's something we'll be learning in the next few years. It's more active, 10 times more active against HIV-2 than other NRTIs. Whether that has any clinical practical benefit, nobody knows. It has a long half-life for its intracellular triphosphate, greater than 72 hours in rhesus macaques, similar to or longer than tenofovir diphosphate. It also, because of its plasma pharmacokinetics and slow elimination, it has the possibility for once-weekly oral dosing. And it could be dosed less frequently than that orally. So when you do surveys of patients and ask them, if you're tired of taking once-daily pills, what would you like to switch to? Taking once-weekly pills actually pops up near the top of patient preference surveys. And so I think we ought to be paying a lot more attention to less frequent oral dosing, and a drug like this gives us that possibility. This drug can also be incorporated into implants, and I'll show you in animals, the data suggest that implants with EFDA or MK8591 could have dosing intervals of greater than a year, which is very exciting. So these are data that were published last year by Stephanie Barrett and her colleagues from Merck, looking at 3 different implant formulations of EFDA. The formulations in A and, The formulations in A and B the formulations in, I think the formulations in A and B are bioerodible, and the formulation in C is inert, more like the TAF implants. And what you can see is that with one of these bioerodible implants, and the PCL, this is the polymer that's the basis for the implant, this drug releases antiretroviral concentrations of EFDA for more than a year. In fact, for almost 2 years. The time down here is in days, out to 600 days. So this is a remarkable pharmacokinetic profile for a product after the implantation of a single implant in laboratory animals, and we're going to be seeing a lot more about this drug in the next few months, including at the meeting coming up this week. Right now, this kind of an implant is most likely to be used for prevention because there's questions about what you would partner it with. You could do an implant and then do injections of cabotegravir or a broadly neutralizing antibody, but right now its first clinical development is likely to be for prevention as a single agent. Okay, I'm going to finish with some technology that may seem like science fiction. I think it's not. I think we're on the verge of applicability of these technologies to humans. This is transdermal drug delivery, so-called microneedles. 20 years ago, when I was first in the business of HIV drug development and talking about novel ways of drug delivery, people would ask me, "Are we ever going to have the antiretroviral patch? You know, you just smack it on your forearm after you take a shower, go about your business, come home, rip it off in the morning, and replace it with another patch." And I said, "Nope, we're never going to have that because our drugs aren't potent enough." Well, that's changed. Now our drugs are potent enough, and we have transdermal drug delivery technologies that I think really can deliver antiretrovirals in a practical way for both treatment and prevention. And this has real interesting implications for infants and children. particularly infants, where drug patches can be easily applied by a mother in the middle of the back and can't be removed by the infant. So very, very exciting potential there. Okay. Not sure if you're laughing because many of you have been parents or if you're laughing because many of you have been infants. This is an electron micrograph of a variety of microneedle patches. These are synthetic, but they're generated in these microforms. But there's incredible versatility in how you make these products. And so the products that you're looking at here, there's two different colors in the needle, and that's because it's two different drugs. So these needles actually have two different substances embedded in them. You can make micropatches that deliver multiple drugs simultaneously. Their initial clinical application has been for vaccine delivery. So there have been clinical studies of microneedle patches for delivery of vaccines, which is great in areas or for people who don't like the idea of an injection, either intramuscular or subcutaneous. But there is now a development of microneedles for drug delivery. So what's it like to wear a microneedle patch? This is me. I was at a meeting in November on long-acting drug development, and several of you were also at this meeting. And there were representatives from a company that is making microneedle patches, and they were passing out these placebo patches. At least I hope they were placebo. So this is my hand. It's the same watch. Oops, go back. It's the same watch, it's the same freckles, it's the same everything. I slap this patch on in the morning. It kind of feels like putting on a piece of sandpaper. It's like taping a piece of sandpaper to your skin. It's not, it doesn't bother you, it doesn't irritate you. I wore this patch the entire day. And at the end of the day, I went back to my hotel and I said, okay, now I'm going to take the patch off and I'm going to take a picture of the side effects. And I was expecting some redness and some swelling. And when I removed this patch, I saw nothing. So this is the equivalent of thousands of mosquito bites without the itch. These needles are so small, you do not feel it when they puncture your skin. And when they come out, you see essentially no side effects. They don't cause bleeding. They're not large enough to cause bleeding. They're not large enough to injure capillaries. So this is a really exciting new drug development technology that has particular application, I think, for infants, children, and adolescents. Although adolescents, you worry about them taking the patch off if they don't want their friends to notice they have it on. But certainly for infants and children, I think this is a great leap forward. There's one of the most interesting applications we've been working on is to use microneedles to deliver nanoparticles. So you actually make the microneedle out of nanoparticles of an antiretroviral drug. The patch is put on the skin. You leave it there only for a few hours or maybe a day or two. The nanoparticles pass through the skin into the subdermal space. They form a reservoir in the subdermal space that you can't even feel. And those nanoparticles then dissolve slowly over several weeks, as is the case for cabotegravir and rilpivirine. So this is a way to deliver a drug like long-acting cabotegravir or rilpivirine without ever having to sterilize needles, go to the clinic, any of that. You could go to the pharmacy and pick up one of these and apply it yourself. So I think this is great technology. It is in the early phases, but this is a situation where I think the technology is there, the need is there, and putting those two things together, I think we are going to see clinical studies of these kinds of devices in the next 2 or 3 years. We have done some modeling exercises with cabotegravir in such a patch to see how large a patch you would need to deliver a dose of cabotegravir for 28 days. equivalent to and produce concentrations equivalent to what's currently being produced with intramuscular cabotegravir given every 4 weeks. And so these data look at several different patches with different drug concentrations and different drug delivery rates. And the best of these patches can deliver cabotegravir at antiretroviral concentrations for 4 weeks with a single application with a patch size of as low as 30 square centimeters. Now, 30 centimeters square may sound large to you, particularly maybe large for an infant or a child, but that's about the same size patch as the original nicotine addiction patches, the so-called Nicoderm patches. Those patches were about 30 square centimeters put on the arm. So now we're beginning to get into the realm of practicality and feasibility for this kind of a drug delivery device, and cabotegravir is not the most potent drug we have out there. If we could do microneedle patches with the Gilead Capsid inhibitor or MK-8591, I think you'd see much, much, much smaller patch sizes, maybe down to the size of that little circular patch I had on my hand. Okay, so I want to finish with how are we going to do this for infants, children, and women who become pregnant. I want to share with you some of the main recommendations of a consensus conference we sponsored in November of 2017. These are the principals who were the organizers of that conference. Many of them are in this room, so thank you for being a part of this activity. This conference was organized and sponsored by the Longacting Extended Release Antiretroviral Resource Program, LEAP, which is a program that we founded with help from the NIH 5 years ago to facilitate development of long-acting and extended release formulations. And the results of this consensus conference were published last week in Lancet HIV, so you can read all about it now. And I want to acknowledge Sharon Nachman, who is the first author on this publication. So what are the key recommendations for neonates? Well, first of all, neonatal washout studies describing the disappearance in the newborn of drug present at birth following in utero exposure are necessary, followed by dosing studies to describe safety and pharmacology in neonates and young infants in order to allow use of novel formulations for neonatal and young infant prophylaxis and early treatment. And so the idea here is mom is on a long-acting formulation. The neonate is going to have concentrations of that drug or those drugs, and we can measure that in washout studies and get some sense of neonatal pharmacokinetics, and then actually use those data, model those data to create dosing strategies of long-acting formulations for the neonate. What about children, pre-adolescents? Well, protocols for trials of long-acting and extended-release formulations in children should be developed alongside adult trials, not after adult trials. And pediatric studies should be initiated as soon as early safety data from adult trials are released. Pediatric clinical trials should simultaneously enroll all pediatric age and weight cohorts, rather than sequentially enrolling cohorts of decreasing age or size, which is the way we've done it in the past, and as you all know, that is a painfully slow drug development process. Long-acting formulations for children should be harmonized across weight bands. As I said at the outset, I don't think that's magic. I think that's simple. We should be using the same formulation for all humans. The only thing we need is a model to tailor the dose to the individual being treated. And with these kinds of formulations, that's very straightforward. And finally, what about adolescents? Adolescents should be included in adult clinical trials or in separate trials proceeding in close proximity. Strategies to maximize recruitment and retention of adolescents in clinical trials should be employed, and this is really to avoid adolescents being left behind. If adolescents down to age 10 or age 13 are enrolled in adult clinical trials, they're not going to be left behind. And that's particularly true in the international setting, and we know that's feasible because it's been done. So in general, acceptability studies should be conducted early in the development of these formulations to understand the attitudes of pregnant and breastfeeding women, children of different ages, their parents and care providers toward novel drug delivery systems. There have been several of these surveys done and published recently. We published one in JADES last year. Coordinated action, and again, generally speaking, adolescents in particular want these formulations. They love the idea of not having to take pills every day. They don't even mind coming into the clinic to get an injection if it means they don't have to think about it for 4 to 8 weeks. So I think this is a way to get adolescents back into treatment. Coordinated action involving all key stakeholders is required from the early stages of development to maximize the chances of success. We need to work with our pharmaceutical partners to make this happen, but we need to indicate to them that we know we can do it, and we have a way to do it, and we have a way forward. And we're not going to ask them to develop pediatric-specific formulations because they don't need to in general. And so, you know, the future is now. It's time for us to act, and it's time for us to do something about this because it's just taken too long. So I want to close by thanking all my colleagues who've loaned me slides, published data, and unpublished data. I particularly want to commend you to the LEAP website, longactinghiv.org. It is a compendium of very useful information for those of you who follow this field and are interested in this field. We have Mark Morachnik on our executive committee. We are proposing to have Elaine Abrams on our executive committee. So pediatric formulation development is a big deal for us going forward. And I want to thank you all for inviting me this morning. Thanks.